Okay, we're back. For the back for the second part of chapter 4. There's a bunch of other stuff in chapter 4 also, but we're just going to look at the cell part and gene and gene function of gene and gene function. Okay. DNA. Well, that's what genes are made out of DNA, deoxyribonucleic acid, right? This is the hereditary material, the genetic material for all life on Earth. It's a double helix, has a phosphate sugar backbone, which is always the same. That's not where the information is found in this informational macromolecule. It carries information. The information is in this series of nitrogenous bases that are attached to the sugar phosphate or phosphate sugar backbone. It's called base pairs. Remember, there are 4 nitrogenous bases in DNA: adenine, thymine, guanine, and cytosine. They all begin with a different first letter, so we just use the first letters, A, T, C, and G. And so remember, it's a double helix. There's 2 strands, and they're around each other. And the nitrogenous bases on one strand are linked to nitrogenous bases on the other strand by hydrogen bonds. And the base pairing rules, also known as Chargaff's rules, because Dr. Chargaff was the guy who figured this out a long time ago, that A always binds with P with 2 hydrogen bonds. C always binds with G with 3 hydrogen bonds. And so an AT, TA, CG, GC called a base pair. Okay, eukaryotic chromosomes. They are, one thing is they're linear. They have a beginning and an end, as opposed to prokaryotic chromosomes that are circular. They're wrapped around histone proteins. Prokaryotic chromosomes are not wrapped around histone proteins. They're naked DNA. But eukaryotic chromosomes have histone proteins. Oh, and something I forgot to mention in the other lecture on cells and cell organelles, that's going to come up in group activity 1, which I want to get to next week, is the fact that we have chromosomes in our cells. These eukaryotic chromosomes are in our nucleus. There's always an equal number of them. Well, we have 46 chromosomes. It's equal. You know, if you're an onion, you have 16. If you're a fruit fly, you have 4. It's always an even number. And this is because we get those chromosomes from our parents. We got 2 parents, and they both give us the same number of chromosomes. So it's always divisible by 2. It's always even. However, that's not the only place in your cells where you find a chromosome. You also find them in mitochondria. Mitochondria actually have a chromosome in them. And the really unusual thing is that it's a prokaryotic chromosome. It's circular and it's naked. Also has other characteristics of a prokaryotic chromosome. It's a bacterial chromosome. So what on earth is a bacterial chromosome doing in one of our organelles? Well, here's the theory about how that happened. It's called the endosymbiosis theory. I should have mentioned it in the last lectures, where it's thought that mitochondria used to be an ancient bacteria, independent bacteria. And about 1 billion years or so ago, this large eukaryotic cell engulfed this smaller bacterium, maybe intending on eating it, but instead of eating it, they made a deal. They engulfed it, and because this is where cellular respiration takes place, most of cellular respiration takes place. A lot of ATP is made. Remember, it's the powerhouse of the cell. So a deal was made. Let's work together. I make ATP, and you protect me because you're bigger and badder than I am. And so this is where two organisms live together, called symbiosis. Endosymbiosis is an internal cellular symbiont, this ancient bacteria that now has become our mitochondria. It sounds bizarre, but it's the best idea, best solution to how a bacterial chromosome got in our mitochondria. And the thing is, your nuclear chromosomes, the eukaryotic chromosomes, they came from both parents. Your mitochondrial chromosome only came from your mom. That's it. You don't get, you get your mitochondria from your mother, not from your father. Sperm have mitochondria in the meat piece that fuel the flagella, but they don't enter the ovocyte on fertilization. Only the sperm head does. And in the ovocyte are obviously mom's mitochondria. Okay. So I mentioned, so that's good to talk about it now. I meant to talk about it in the last lecture on organelles. Okay, replicated chromosomes consist of two sister chromatids connected by a centromere. Okay, single chromosomes, one is double-stranded molecule. Before cells can make other cells, they have to replicate the chromosomes. And they do this. I'll show you in a later slide how this is done. And you end up with a chromosome that looks like sort of an H with two DNA strands called chromatids now. And they're called sister chromatids because they're absolutely identical, genetically identical, unless mutations happen. We'll get to mutations. And they're connected by a centromere. Okay. And there is a chromosome, and that's H-shaped pattern. See, those are called sister chromatids. And there's the centromere holding them together. And these things are found in the nucleus. Here's the nucleus, that's connected to the rough endoplasmic reticulum, functions of the nucleus, cellular regulation, houses DNA molecules that serve as the genetic instructions for synthesis of proteins. So remember, the information in a gene is a blueprint to make a particular protein. It codes for the primary sequence, what amino acids go in what order to form a particular protein. That's what most genes are. And notice that the blue is the DNA, and it's wrapped around these orange histone proteins in supercoiled. And there is a double helix right there, and there are the base pairs and the hydrogen bonds, 3 between G and C's, 2 between A and T's. And then there's in a gene is a promoter region that tells the RNA polymerase when this information is being put into RNA where the message starts, and then a terminal region where the message ends. Production produces ribosomal subunits and nucleolus and exports them into cytoplasm for assembly into ribosomes. So actually, there's a little area called nucleolus inside the nucleus where a couple things happen. One of which ribosomes are produced there and then put into the into the cytosol or in the rough endoplasmic reticulum, but also something called RNA processing happens there, and we'll talk about that later. Okay. So chromatin is the stuff you find inside the nucleus that consists of DNA wrapped around these histone proteins. Okay. So genes are found in a linear array along chromosomes in loci. So gene locus is where a gene is located on a chromosome. And we have 23 pairs of homologous chromosomes. I'm going to get to that in a moment. So you could say that the gene for X protein is on near one end of chromosome 8 or something. So loci is plural for locus. And it's thought that human beings need around 30 to 35,000 genes to make and maintain a human being. You need 30 to 35,000 genes, therefore 30 to 35,000 loci are on those 46 chromosomes. Genes are an information-containing region of chromosome that leads to the formation of an RNA molecule, which may lead to the formation of one or more specific proteins. So we're going to get down into what's called protein synthesis, and actually you can call it gene expression. What happens when you express a gene is that the protein that it codes for is made, and there's two processes that are going on there. One is called transcription, happens in the nucleus, where the information from DNA is put onto a copy of an RNA molecule, and then translation, when the RNA molecule is brought to a ribosome, where the protein that it codes for is produced. The human genome. The human genome is the entire sequence of base pairs along all of our chromosomes. And it has been sequenced. We know what it is. And now the job is to find all of those 30 to 35,000 genes that are found along there and find out what each one of them does. This has already been achieved for much more simpler organisms. There's one nematode worm that has about 3,000 genes, and it's absolutely genetically known. They know where every gene is and what every gene does, and that's what they like to find out for human beings. But it's a little more complicated. Okay, protein synthesis. So this is making proteins from the information found in a gene. And the information on making a protein is found in the series of nitrogenous bases on one of the strands in a DNA molecule. And but it's in a coded form. It's called a genetic code. So it's like a language. So it's like the language of DNA and the language of protein, and they're different. And so that's why when you copy make a copy of a gene on an RNA molecule, it has to go to a ribosome and be translated. So think of it this. I like this analogy. Think of the chromosomes in your cells as like books in a library. And every page, like in a book, every page comes after one another in a linear array. And every page is a recipe for a protein, right? So, so in order to make that recipe, to acquire the recipe, you have to go into the library, open the book. So so you have to go into nucleus, unwind the strain of DNA and make a copy of one of the strands. So it's like making a copy of the page. So this is not a lending library. All you can do is make copies of the pages. So make an RNA copy of the page and you take it out of the library. And but the page is a different language. So now you need a translator, and you have to translate it in order to follow the recipe and make the protein that it codes for. Okay, and so that code is called a genetic code. So transcription, as I mentioned, is making that copy, that RNA copy of one of the strands of the DNA. So actually only one, it's a double helix, and you gotta unwind it, and you only copy one of the strands. That's called the coding strand. So only one strand actually has the information, the other strands just stay to hold things together. And so you have to make an RNA copy. Now RNA is a little different than DNA. For one thing, it can be single-stranded or double-stranded. And there's different types of RNA. What we're making here in transcription is called messenger RNA. And it's called RNA because it's ribonucleic acid. So it has sugar called ribose in its backbone, not deoxyribose. And the other thing is it has 4 nucleotides, but one of them is different than DNA. There's no thymine in RNA. There's something called uracil. So A binds to U in RNA. It doesn't bind to T. So U takes the place of T in RNA. Okay. So the first thing that's made is called a pre-mRNA or primary transcript. Right? So that's a copy of one of the DNA strands. And this takes place in the nucleus by a protein called, an enzyme called RNA polymerase. Then this primary transcript has to be processed in the nucleolus. This is called RNA processing. And this was truly bizarre when this was discovered, in that genes, eukaryotic genes, are composed of what are called introns and exons. Exons are meaningful strands of DNA. Introns are nonsense strands. It doesn't code for anything. It's strange. So eukaryotic genes are discontinuous. It's as if, you're reading that book and everything makes sense. And then all of a sudden you come to an area that's gobbledygook. It's all letters, but they're not forming words or anything. It doesn't mean anything. And you go along and see all this gobbledygook until finally, starts making sense again. And starts making sense from where you left off over here. It picks up over here. In between is gobbledygook. Those are called introns. And they have to be excised before you get a mature mRNA molecule that can go out to a ribosome. This is called RNA processing, and it's done by a very ancient enzyme called a ribozyme. And this is a bizarre enzyme because it's not a protein. It's actually a type of RNA molecule. It's an RNA enzyme. And this was really surprising to figure this out. When people figured that out. And it's thought that it's the holdover from, a billion years ago or so ago, or actually more than a billion years, like maybe 3 billion years, where at one time it's thought that proteins really didn't exist. It was RNA, it was just nucleic acids. And RNA molecules did the enzymatic things. So enzymes are crucial for life. You have to have enzymes to speed up chemical reactions. But at one time it was thought that enzymes are RNA molecules. And then somehow proteins were invented. And that sort of makes sense because this is a really complex thing, RNAs and proteins. And you think it would be really hard to believe how both of those could have evolved at the same time. So one of them evolved first the RNA enzyme, and then the enzymes were somehow invented. And they seem to do enzymatic functions better than the RNA. And so all the RNA enzymes are replaced by proteins, except this one, the ribosome. It's still there. So it's a very, very ancient enzyme. Okay, so then you have a mature mRNA. It leaves the nucleus by one of the nucleopores, goes to a ribosome. That ribosome could be one of the free ribosomes in the cytosol, or it could be attached to the rough ER. All right. So then we got to do translation. This is happening at the ribosome, and it so you have to translate the language of nucleic acid into the language of protein. Now, so it's sort of like a language, because then what things do you need for a language? Well, let's say what things you need for a Western language. Not the Eastern languages don't have alphabets, but the Western languages has an alphabet. So we have an alphabet, and how many letters are in the alphabet for nucleic acid? 4. It's only 4 letters. A, T, C, and G. Or an RNA, A, U, C, and G. And then you have to have words, and they be strung together into a sentence. And we're going to find out that the words in nucleic acid language are always just 3 letters long. And then what else do you need to make a language that you can read? You need punctuation. You need a capital letter at the beginning of the sentence, a period at the end. And there are things in the genetic code that are punctuation for the genetic code. Now, for amino acids, they have 20 letters in their alphabet. Right? There are 20 different amino acids that can be put into proteins, and those words can be any length. Small proteins are 10, 15 amino acids to hundreds of amino acids long. Okay, now a translation, you have to use what are called tRNA molecules, a transfer RNA. These are small cloverleaf-shaped RNA molecules that have a binding site for an amino acid on one end and what's called an anticodon at the other. And then first I'll define codon first, and then later I'll talk about anticodons. Codons are these 3-letter words that are found in the genetic code in the nucleic acid language, and an anticodon binds to it. Okay. And so there's 61 different types of tRNA molecules. Well, I'll show you where I get this number. Okay. Here is the genetic code. Now, I have to, memorize this, but here's the thing. So I said it was the information in DNA is in coded form. Well, what's the code? So it couldn't be a 1-letter code because there are 4, remember there are only 4 letters in the nucleic acid alphabet, and there are 20 different amino acids. So it can't be a 1-to-1. How about a 2-letter code? Well, if you had 4 different letters, in each one, how many different 2-letter combinations can you make? If you have 4 letters, you can make the combinations with 16. 4 to the 2nd power. And that's not enough. we got 20 amino acids. So the code is a 3-letter code because there are 64 different ways you can put 3 letters together if you start with a choice of 4, right? So it's a 3-letter code, and there's 64 different what are called codons, 3-letter combinations that code, or most of them code for an amino acid. And since you only need 20, but you got 64, it's called a redundant code. So most amino acids are coded for by more than one codon. And you can see here. So I can pick any, so how you read this chart here is the first letter is on the left, the second letter on top, third letter on the bottom. So like CCA, there's a codon. What amino acid does CCA code for? All right. You go over here, first letter is a C. So it's got to be in this row right here. Second letter is a C. So it's got to be in this column here. So it has to be in this square here. No, what am I talking about? This square here with 2 C's. I said CCA. There's CCA right there. What's it code for? Proline. So I can give you any 3-letter combination. Now we use U rather than T because this code is in RNAs, converted to RNA, and RNA uses U rather than T, so that's why they're used. So, and notice that proline, all 4 of these codons code for proline. So I think the maximum is 6. Some amino acids are, I think it's like serine has 6, yeah. Some have 4. One of them only has 1 special one. This one right down here, AUG, codes for methionine. But it's something else. It's also the start codon. It's the capital letter that tells the ribosome where the beginning of the message is. All right. It's but it could be found inside of the message too. It could be found inside the protein, where is a methionine AUG. It's slightly modified to show that the ribosome, that this is the AUG that starts the message, is not found inside the message. It's the start codon. So it's 1 start codon, AUG, slightly modified. And then there are 3 codons that don't code for proteins. They code for the period at the end of the sentence. They are the stop codons. And it's up here, AUU, AUG, and UGA. They are stop codons. They tell the ribosome, okay, stop, you're finished. The protein is complete. Okay, and this is the transcription, and this is right out of the book in chapter 4. How you have to unwind the strand, then you have to have RNA polymerase come along, and if it sees a G on the coding strand, it puts a C. And if the next one were T, it would put an A. If the next one would have been A, it would put a U. You see how it's over. So it's a complementary strand complementary copy of one of the strands of DNA. And once that hits the terminating region, it comes off the DNA. The DNA is put back together again, and the page is, and the book is closed and put back on the shelf. Then that pre-mRNA strand, sometimes also called a primary transcript, is right there. And notice, see, it has exons, introns, exons, introns. Remember, now that has to go to the nucleolus for RNA processing, where the ribosome will excise the introns and splice together the exons. to make a mature mRNA molecule. So we have initiation, elongation, termination. Now this, it's really small here, but you can see right there in the book, is how this message, now the mature mRNA is translated at the ribosome. There are two sites where what happens is, you come along. So actually, there are two parts of the ribosome, the big subunit and small subunit. They clamp on both sides of the mRNA, right with the AUG, the start codon is. And so we got to start with methionine. So how is methionine brought? It's brought by a tRNA. So it's a tRNA that has a binding site for methionine on one side, and on the other side is a 3-letter code called an anticodon that's going to bind to the AUG. And you know what that 3-letter code is for that anticodon, because of Dr. Chargaff. AUG, what binds to AUG? UAC. Right? Because A binds to U binds to A, G binds to C. So the anticodon for methionine has to be UAC. And that binds. And then the ribosome moves along, goes to the second codon. Let's say the second codon is UUU. That, I happen to know that codes for the amino acid phenylalanine. So then another tRNA comes along that has a phenylalanine attached to one end of it. And it has an anticodon. What's its anticodon? AAA, because that binds to UUU. And then that's put in the second that's attached to ribosome. Now, as an enzyme comes along and attaches the AUG to the phenylalanine, and then the ribosome moves along to the third codon. That third codon could be CCA. And so we already know that's proline. So there's another tRNA that carries a proline, binds site for proline. It has an anticodon, and if it was CCA, it has to be GGU, the anticodon, and so on. And so the ribosome moves along to every codon in place, and a different tRNA brings over a particular amino acid. Now these tRNA, they're made in other places, and the amino acids are put onto them in different other places. And so this is why I was saying there has to be 61 different tRNAs with a different anticodon, because there's 61 codons that code for amino acids, so you're going to need an a tRNA to bring that amino acid over. Because remember, there's 3 stop codons, and they don't code for it, except for termination. You see at the right-hand side here, there's going to be a stop codon, and this release factor is going to bind, and that's going to break apart the ribosome, and the protein will drift away. Oh, remember, so after we mentioned the AUG and the phenylalanine were linked together, then we went to CCA, there's a proline, the proline's attached to phenylalanine, and so on and so forth all the way down the line. So then that protein comes off, the ribosome folds in its particular shape, and either does its job or goes into the rough ER and then onto Golgi apparatus to be modified or whatever. Right? So then we have the completed protein. So gene regulation and expression. So gene regulation is, when do you do this process? When do you make the protein? And making a protein is called expression. And gene regulation is, when do you do this? And there are and it's really complicated. Sometimes the binding of a hormone to a cell will turn on a gene, for example. Genes for all proteins found in all cells. So you have to have a gene for every single protein that you make. But here's a conundrum. We can make about 1 billion different proteins, but we only have about 35,000 genes. Well, how is that possible? Well, this is the thought behind where this intron and exon things comes about. If you can, you have a certain X number of exons, let's say you have 100 exons in a gene, and you can mix and match the different exons. So you have, so you take exon 6, 14, 27, 34, 67, 89. You take those, you put those together in mRNA, you make one protein. To make another protein, you take exon 2, 13, 34, 71, 94. Take those exons, put those together, you make another protein. So genes can code for numbers of different proteins depending upon the mixing and matching of the exons. It's like, let's use the example of poker. So you have 52 cards in a deck, and then you deal out 5 cards. How many different hand combinations can you make with 52 cards? I don't know, I probably could figure it out, but it's a lot. It's really a lot. So think of it like this. You know, all of the exons are cards, and you deal out 5 of them for one protein, a different 5 for another protein, another 5 for another protein. And so that's how we can make about 1 billion different proteins with only about 35,000 genes. So, oh, and here's an interesting thing about genetic variability. We say we're all genetically unique. You're genetically different than I am. Well, what does that mean, really? Do you have different chromosomes than I have? No. Well, if you're female, you don't have a Y. I got a Y. Females don't have a Y. So that's the only difference there. Right? But you have the same chromosomes I have. Right? Do you have different genes than I have? No, you don't. There are about 35,000 genes to make and maintain a human being, and everybody's got the exact same ones. One exception, again, is the Y. I have a Y chromosome. There are certain genes on there. They're not found on the X. So therefore, so that's one exception to the rule. Males have a few genes that females don't have because they have an X chromosome, a Y chromosome. But all people have the same chromosomes, the same genes. The difference is the alleles. I'm going to get to alleles a bit later on too. Alleles are genes that do the same thing, but slightly differently. Like for eye color, there's a gene that codes for eye color. But one of them codes for brown eyes, one codes for blue eyes, one codes for hazel eyes. You see, so how we are genetically unique is the set of alleles that we have at our 35,000 or so loci. And why do we have different set of alleles? Because we have different parents, and they gave us different sets. Okay. DNA replication, like I said, you have to, before you make new cells, you have to replicate your DNA, have to make replicated chromosomes. And that's done in the nucleus by a molecule called DNA polymerase. In the nucleus. Now, this DNA polymerase is not a perfect enzyme. It makes mistakes. Sometimes those mistakes are corrected, other times they're not. And this is what a mutation is. It's an error in DNA replication. Every time you copy DNA, you're likely to make mistakes. Actually, 3 mistakes on average. On average, DNA polymerase makes 1 error for every billion base pairs that are copied, and there are 3 billion base pairs in the human genome. Okay, and there is DNA replication. In the S phase of the cell cycle, the DNA is separated into single strands. DNA polymerase comes along on both strands, and if it sees a T, it puts an A, sees a C, it puts a G, and makes 2 identical strands from that one strand. It's called semi-conservative replication. That is, the remaining chromatids are composed of 1 original strand plus a brand new, newly made strand. Okay. Okay. On that, and so I'm going to go into alleles again when we get to, when we pick up on the next video, because we've already gone about a half hour. So I'm going to close this one. This will be 203 gene 1. And catch you on the flip side here.